Hey, hello everyone. Thank you for attending the final exam. Today I'll be covering my thesis topic, which is about statistical modeling for a specific type of adaptive evolution using identity by descent segments. And the identity by descent segments will be our data. The agenda for today is that I will first introduce the problem, so give some motivation about adaptive evolution and define identity by descent. Next, I'll cover project 2 of my thesis, which is on establishing a central limit theorem for a sample mean of correlated binary random variables, which in our case will be the identity by descent rate. Next, we'll build upon our central limit theorem to develop a multiple testing correction for an IBD-based selection scan. That's my third project. And I'll briefly touch on the content in my general exam or my candidacy exam, which was about modeling recent and hard selective sweeps with the IBD data. For topics 2 and 3, we'll have some motivation, technical details, and empirical studies. I'll have some real data analysis results for topic 4. Okay, the motivation. So we're going to consider this species, the peppered moths, in circa the 1900s. And we're in Britain, and at that time, say the white color phenotype was more frequent in the population of moths, but we're in the early 1900s. It's the beginning of the industrial age. There's a lot of heavy machinery. And so there's more pollution in the environment right now, this change in the environment. And so what might have happened, or one interpretation of what happened, was that the black color phenotype increased very rapidly in a few years to a few decades. And the geneticist's interpretation might be that this was a phenotypic advantage, and alleles that were associated or mechanistically driving that phenotype would also increase in frequency. We're going to use long identity by descent segments to model such a scenario. I'll define this, and it makes the most sense in the context of a pedigree. So on the right side of the figures here, that's the past. And then on the left side of the figure is the present. And so we see in the past, we have these two founding individuals. They're diploid organisms. So they have 2 chromosomes, which are given with offset colors. When they have offspring in the next generation, there's this process in genetics called recombination that will exchange DNA across the different chromosomes. This creates a mosaic of the chromosomes in the next generation. And so if we keep doing this recombination process for many generations of offspring, and then we look at the current samples in the current day, and so that's the far left set of chromosomes, these long stretches of DNA, identical by descent from the founders, may still be preserved. And so I'm denoting this with this little dot that's attached with this vertical gray bar between 2 different chromosomes. So if this identical by descent stretch of DNA is long enough, there are modern algorithms that could detect these very accurately. So this diagram is actually coming from Ancestry DNA, a commercial company similar to 23andMe, who might be able to give you results like this so you can learn more about your distant family. And we also have academic software that can make these reliable inferences. Throughout this talk, I'm not going to get into the details of these algorithms. We're just going to assume that the IBD segments and their lengths we have faithfully detected with quite a high amount of precision. Okay, And now I want to talk about a specific ancestral tree process at a locus on the genome and how that's related to identity by descent. So at the bottom of this tree, I have in dark blue the current samples, and then higher up on the tree, I have in light blue their common ancestors. And we model the time upwards on this tree, which is the time to the common ancestor, as an exponential random variable with varying rates depending on how high up you are on the tree. And this model from Kingman in the early 1980s is well motivated as a continuous time limit of this process where we have random mating, and then these children have a parent that you can draw from as you're doing that process backward in time. It's referred to as the Wright-Fisher process. And so an explanation for a distorted tree on the right is our example of very rapid selection, referred to sometimes as a selective sweep. This is one model for it. And so we now have these red and orange dots. The red ones are the current samples with the selected allele, and the orange ones are the ancestral samples with the orange allele. And at our horizontal bar that's orange, this is where a mutant was maybe introduced or became advantageous due to an environmental change. And because of that advantage, we see that those in the present day with that allele have more recent common ancestors. Those common ancestors are closer to the present day. And all of this together is that there's been less time for the recombination process to act. And this means that these stretches of DNA that we got from, say, the founder of this mutation has not broken down or shrunk as much. And so we might be able to detect this phenomenon of these long stretches of DNA shared identical by descent. The primary aims of this thesis, it's a statistics thesis, is to study rapid positive selection using the IBD segments. And we want to bring a lot of statistical rigor. So for me, this means that we're going to propose a multiple testing threshold when you're doing hypothesis tests across the genome that will control the family-wise error rate. The family-wise error rate is the probability that we make one or more rejections of a null hypothesis when the null model is in fact true. The other case is that we will provide an estimator for the selection coefficient that will have desirable properties. And this was the main topic of the general exam. The selection coefficient is this parameter that defines the expected rate of allele frequency change in our model of rapid evolution. We did discuss topics about asymptotic consistency and sufficiency in the general exam, but I've predominantly been interested in confidence intervals that have the proper coverage. So if you give me a 95% confidence interval, we expect to see in 95% of simulations that our confidence interval contains the true parameter. Okay, so after setting the scene, I'll transition to my second thesis project, the asymptotic normality, the large sample normality of the detectable IBD rate. Up in the right-hand corner of this slide, we have the classic bell curve, the standard normal distribution that we may be familiar with from our first statistics courses. So we have this theorem called the central limit theorem that if you collect a bunch of independent data observations and you take their sample mean, this should converge to the standard normal distribution. It's not this simple if you actually have correlated data. In our case, we have this complex correlation process both in space across the genome, but also back in time with our coalescent process. So our aim here and our achievement is to establish a central limit theorem for this specific type of correlation. I'll define our sample mean here. So I'm considering 4 haplotypes at a locus. The locus is here, these singular dots. We're going to model these identity by descent segments. Sorry, I don't know why that's flipping. We're going to model these identity by descent segments to the right and left as independent exponential random variables, and their rate is going to depend on the common ancestor time. So this is the modeling of our recombination process, and it's been known from almost a century, really, from the work of Haldane, that the Poisson process model is a good model in this case for the recombination process, and that is sort of where these exponential random variables are coming from. Here I'm using this unit, which I call W centimorgans. The morgan is the distance between 2 points on the genome for which we expect to see one recombination event in one offspring gamete. And then centimorgans is analogous to centimorgans in centimeters and meters. So our W's are the identity by descent segment lengths that are shared between, say, labeled haplotypes AB, AC, BC, CD, and so there's 4 choose 2 here, 6. These are long with shape 2. And we will have our identity by descent segment indicators, the Y's, that will be either 1 or 0, depending on if the IBD segment lengths exceed this W threshold. And at the end, we're interested in the sample mean of correlated binary variables, which is the sample mean of these IBD segment indicators. Our central limit theorem is going to depend on 3 parameters, but one in particular will be important, and this is the centimorgan detection threshold, W. I'm going to compare this IBD rate distribution at a locus to the binomial model. So the binomial model concerns independent binary observations. And our binomial model here will be using the success probability that for a single IBD segment is approximately 1 over the W centimorgan detection threshold times the population size. And so that's the success probability I simulated under. When we look at the differences between the binomial model and the IBD rate distribution, what's immediately obvious is that this IBD rate distribution has more covariance than its independent analog, the binomial. Here I've been using this 2 centimorgan threshold. As I increase our centimorgan threshold to 3, our blue and orange kernel density estimates are starting to look more similar. And here, comparable covariance. So this is giving us some intuition that one situation in which perhaps we're going to achieve the central limit theorem is using a large centimorgan detection threshold. Okay, here is slide 10. This is the most important slide of this talk, and we're going to go through this quite slow. I call it the seminal theorem of the talk because everything I'm going to tell you henceforth is going to hinge on this and build upon this. So under the following interpretable conditions, the standardized identity by descent rate at a locus is standard normally distributed. The first condition is that we have a large sample size, little n, that we have a large population size, big N, that is scaled by the centimorgan detection threshold W. The big N, the population size, is considering this idealized model where you have randomly mating haploids, non-overlapping generations, no population structure, etc. And so these are conditions that you can look up that are common in population genetics. The second and third conditions will be the most important. The second one is that we have population size that is much smaller than sample size squared. When we're considering IBD segments, these pairwise IBD segments, that's going to be an n choose 2 number of binary observations. So the number of observations we have is this sample size squared order. And what this is saying is that we need to have enough data. We need to observe at least enough ones to establish a central limit theorem. And if, say, the population size is quite large, we might have a very rare probability of observing an IBD segment. The third condition is the most important. This is that the sample size is much smaller than the scaled population size n times w. What this condition is about is vanishing covariance terms, such that these IBD segment indicators are not too correlated with each other. I'll just comment for the technical audience of, say, mathematicians, that conditions 2 and 3 are about little o statements, and that our proof is going to follow from a result, or it's going to appeal to a result in Chandrasekhar et al. 2023, where they give moment conditions for a general covariance-based central limit theorem. Okay, so let's grapple with the covariance structure. There will be three covariance types that I'll talk to you about today. These are the only three. So we have the covariance of the IBD segment indicator between haplotypes AB and the IBD segment indicator between haplotypes AB. So in the context of the coalescent tree, I've circled the relevant branches and common ancestors and samples here. So these are the orange and green circles, and they're containing the exact same IBD segment indicator. There will be approximately n squared of these covariance terms when we're considering the sum of all covariance. And each of these marginal covariance terms will be of the order big O NW to the -2. Now we're considering the second covariance type. So we've got the IBD segment indicator AB with the IBD segment indicator BC. So I've changed this green oval such that it now contains the connection between the current samples B and C. And we'll note when we look at the intersection between the orange circle and the green circle, there is a common branch length, and there's also a common sample. There are going to be approximately n cubed of these covariance type terms, and each will be of the order big O NW to the -3. Our final type is the covariance of the IBD segment AB and CD. There are going to be approximately n to the 4 of these terms. And there are here, in fact, 2 possible tree configurations. So I'm showing you one of them. And in practice, to compute this covariance, you'll need to integrate over the 2 possible coalescent tree types. And for all of these covariances, we need to integrate over the branch lengths of the coalescent process and integrate over the recombination process to the left and right of the focal location. Each covariance of these types will be of the order big O NW to the -4, and there will be n to the 4th power terms approximately of this type. Okay, so now we've grappled with what these covariance types are, and now we'll just build some intuition for that third condition and how that will imply some vanishing covariance terms. So on the left, we have a, say, typical coalescent tree that could have been observed, say, or might have happened. And this is coming from the population of here, just nominally 15 individuals. I have this vertical time axis that I won't give a scale, it's mostly for comparison's sake. And we'll note that these covariances are going to depend on n times w, where n is this population size. So now I'm going to double the population size. And the effect of this on the coalescent process is that the tree on the right is, say, more typical. And the feature of the tree on the right is that it has longer branches. And if we recall, this means that there's more cuts in this recombination process, creating smaller and smaller stretches of IBD data, resulting in smaller correlations. So at least for me as a statistician, it's very exciting to have a central limit theorem to say something theoretically, but we also want to look at this empirically and see if our central limit theorem is in any sense even useful or makes sense. So we developed an algorithm to simulate IBD segments around a locus. I'll say that there were existing tools that could simulate IBD segments genome-wide, but these tools were mostly a secondary feature of much broader simulation frameworks in population genetics, and they weren't necessarily simulating fast enough for the scope of the simulations that we were interested in. So the algorithm that we developed is as follows. So first, we simulate a coalescent tree, and then from that coalescent tree, at each event, we will draw recombination endpoints to the left and right for the haplotypes that coalesce there. We will compare the endpoints of the haplotypes. And if their shared endpoints, their IBD segment, is long enough, we'll keep track of that, since we're interested in how many detected IBD segments there are. We had to speed up this algorithm. The problem, again, is that we're studying pairwise comparisons, and we want to study a large sample size. So this is going to scale n squared. We also want to do billions of simulations to test the distributional properties. In particular, are the empirical distributions that we simulate similar to a normal distribution? To do these speedups, we provided 4 little techniques. I'll focus on 2 of them, and they speed up the algorithm because of probabilistic arguments. The first is pruning. This is when, if a haplotype has its segments to the left and right that are together not longer than our W detection threshold, we'll stop keeping track of this haplotype and making comparisons with the other haplotypes it coalesces with. This is because when you make those comparisons, you're taking a minimum of the endpoints. So we're never again going to observe a long enough IBD segment if the haplotype segment isn't long enough. The second is merging. So if 2 haplotype segments have the same endpoints to the left and right of the focal locus, we're going to treat them henceforth as the same. So we treat them as a class object, if people are familiar with that in computer science, and we can keep track of how many are in this sort of merged group so that we are keeping track of exactly how many IBD segments have exceeded the threshold. Very quickly, I'll just show some computational runtime results. So on the vertical axis here, I have the compute time in seconds. Halfway up the graph is 1 minute. On the horizontal axis, I have sample size in thousands, and I'm doubling this at each tick point. In blue, I'll have our fast method, the solid line. And then in orange, I'll have the orange dashed line, I have the naive implementation. So this is the implementation where I don't use pruning and merging. Here are the results. It's immediately obvious. So this naive method is scaling quadratically with runtime all the way, say, at 8,000 samples. We already have a compute time that's exceeding 1 minute. And this is not tractable for the billions of simulations we want to do. For our fast method, at least up to 16,000 sample size, we are observing some linear behavior that is less than 2 seconds. It is a quadratic algorithm, but our probability arguments are bringing that down, at least by a scalar factor. And I've put a line here at roughly 100,000. This is sort of representative of the size of human biobanks in, say, the 2020s. And so we think that this is fast enough, say, to make these simulations in less than a minute. And that will be somewhat tractable for our simulation and real data analyses. On this slide, I'm going to walk through our main empirical study of our central limit theorem. What the theorem says is that we should reject a null model of normality less often when either the population size or the centimorgan detection threshold increase. We will use a Shapiro-Wilk test. The null model is normal distribution, and the alternative model is that the data is not normally distributed. The vertical axis is the proportion of these rejected hypothesis tests. The horizontal axis is the centimorgan length detection threshold from 1 up to 4. For this, I've fixed the sample size at 5,000 with the confidence level of 0.05. The line plots here, the colored line plots, are showing increases in population size. So from the darker blue to the lighter blues, I'm increasing the population size. What we're observing is that we reject fewer hypothesis tests, which is consistent with the conditions in our central limit theorem. As we move from left to right on the horizontal axis, so we're increasing the centimorgan threshold, we're rejecting fewer tests. Again, consistent with the conditions of our central limit theorem. This darker orange line is a little bit above the confidence level 0.05, but the closest to that of the simulations we did. And possibly counterintuitive to the audience is that when I'm moving from 1 million to 10 million in the population size, we actually rejected more hypothesis tests. This is indicating the balance between our assumptions 2 and 3 in the central limit theorem. So when we had that setting of 10 million in the constant population size, we were observing very few detectable IBD segments. So we didn't really have enough data to do a good sample mean. I'll note that in practice, that centimorgan detection threshold will be fixed by the practitioner, given the high quality of the data to make reliable inferences. And we investigated in our paper when we would reject the null model and what was happening. So typically we had a heavier upper tail to the empirical distribution than one might expect of a normal distribution. We make the discussion point that if one was to use the IBD rate at a locus to do a z-test for selection, that this might give anti-conservative behavior. And so this will lead us into our second project. However, at this time, I'll recap the contribution to the field. So our asymptotic normality result will motivate the method we proposed for multiple testing. And it actually also was motivating implicitly the confidence intervals for the selection coefficient, which I'll mention at the very end. Relative to the existing literature, it's actually usually quite hard to get any asymptotic result in population genetics, as you have to grapple with complicated correlation structures that you don't observe. So if we look at some other authors here, we've got a paper from Shai et al. In 2024 that note that their summary statistic looks normally distributed. Then we have Palomar et al. In 2018 and a Field et al. Paper in 2016 that observed that their summary statistic looks gamma distributed. And so you can perhaps use these observations, these distributional observations, to compute a p-value. But this isn't really the same as having a theoretical reason to believe that your statistic looks like some parametric family. In particular, say for the Shai et al. Case, there are other distributions that look normally distributed, for example, a Cauchy distribution, but that has pathological behavior with very heavy tails. Okay, now we will transition to my third project, which is about a genome-wide significance threshold for an IBD-based selection scan. This selection scan has the following null hypothesis. The null hypothesis is that there is a constant genome-wide IBD rate around a locus, and the alternative hypothesis is that there are sites that will have an excess IBD rate. And we're going to use a z-test, again, motivated by our central limit theorem. This is ongoing work with Professor Sharon Browning, and I will use this language sometimes scan, which for me means that we're doing multiple hypothesis tests along the genome. Contextualizing this with the existing literature on family-wise error rates for selection scans. That's what we're going to talk about on this slide. So there are authors, our group included, that consider, say, you compute a standard deviation and you set your threshold as some number of standard deviations above a median or mean. Some other authors have spaced their summary statistics around the genome and used a Bonferroni correction. And I guess the hope is that they've adequately spaced their summary statistics such that they're not super correlated hypothesis tests. And then some authors in the selection literature have used the genome-wide association testing significance level of 5e to the -8. We'll caution, though, that this actually is based on association tests for human array data in the early 2000s. And so if you're thinking about another species with different correlation structures, longer genomes or shorter genomes, and then sequence data now where you have a lot more markers, the adoption of, say, this nominal significance level 5e to the -8 might not make sense. Altogether, the limitations of these approaches are that we don't know if any of them will control the family-wise error rate. And in none of these cases have we actually done a simulation study to assess if these are valid thresholds, mostly because these methods have been very computationally intensive, either to simulate data or to compute their test statistic. We will actually pursue that. And one of the reasons we can really do this is because our statistic is so simple. It's a sample mean of correlated binary random variables. So recall, we have our coalescent around a neutral locus on the left. And then on the right, we have a coalescent around a selected locus, a strongly selected locus, and that has distorted the structure of this coalescent process resulting in more long IBD segments that may be detected by the modern algorithms. I'll show results for our initial work, Temple, Waples, and Browning, from a preprint in 2023. So on the vertical axis, we have the IBD rate. On the horizontal axis, I have position along the 22 human autosomes. This data is coming from a European ancestry sample group in a United States biobank. The vertical line is our median genome-wide IBD rate. The vertical blue line, the vertical dashed red line is our 4 standard deviations threshold. And I've annotated a few loci where we're exceeding that threshold, and there's been broad consensus in the literature that these loci may be selected for in European populations. We want to improve that selection scan by having a more principled statistical framework for the thresholding. Our approach is going to be to model the Ornstein-Uhlenbeck process, the IBD rate process, as an Ornstein-Uhlenbeck process. This mathematical process has the following properties. So at each point on the process, we have normality. So we feel this is motivated by our central limit in the prior project. We're going to have spatial homogeneity, so common mean, common variance across the genome, and that's a part of our null model. Mean reverting means that if this process starts to drift away from that mean, we expect it to eventually come back. The Markov property is that if you have a lot of observations back in the past, and for your current observation, you're conditioning on them, well, you actually only depend on the most recent observation. And then for the covariance form, it should take, after standardization, a specific form. So we'll have that the covariance of the IBD rate at locus 1 with the IBD rate at its next adjacent locus 2 is the exponent negative theta times delta. Delta is the spacing between our test statistics that will hold constant, and that's user defined. Theta is a decay parameter that's controlling the covariance of the different IBD rates across the genome. And one can understand this as you have these IBD segments that are overlapping some loci, but eventually as you move along the genome, they have been cut and they're not appearing in the next observations. We propose 2 multiple testing methods. I'll emphasize one today. So the first is that we're going to standardize our IBD rates over the genome. So subtract the mean, divide by the standard deviation. Next, I'm going to fit a log linear model. So we'll calculate the covariances between these IBD rates autoregressively across the genome. And then we'll take the log of those covariances, setting it equal to negative theta times the scaled deltas, our spacing parameters. And the theta here is the regression coefficient that we're going to learn with simple linear regression software. And then for multiple testing, we use an approximation method from Sigmund and Yakir's book in 2007. This approximation method is trying to control the family-wise error rate, and it has a term that we interpret as involved in an infinite number of independent tests along the genome with a correction factor for the first hitting time of the Ornstein-Uhlenbeck process. So that's the correlation correction factor. And that aspect of it depends on theta, our exponential decay parameter. So now I have some results for what the family-wise significant levels look like for multiple testing. The first column is the nominal one, so we'll have 0.0 0.01, 0.05, 0.10. And we need to correct for this since we're going to do multiple tests. So the middle column is our genome-wide threshold. And then on the far right, we have the Bonferroni method, where you just indiscriminately divide by the number of tests that you did. And comparing the far end to the middle column, we see that our threshold is at least 6 times larger than the Bonferroni method. It's well known that Bonferroni corrections can be very conservative, which is concerning that we might not have the statistical power to reject the null model when in fact the alternative model of, say, some form of selection is the true model. And again, our method is designed to control the family-wise error rate. Now I have some simulation study results. So on the left, our results, the vertical axis will have the family-wise error rate that we calculated. So we did 500 whole genome simulation studies, and then we tested if at any point the IVD rate exceeded our multiple testing level. So this is going to be the number of at least one rejection over the 500 simulations. On the horizontal axis, we have the significance levels, and then I'll have different colored bars for the different choices of delta, the step size, or the spacing. Here are results. I'm using a horizontal red line as well to aid the audience on what the significance level is. So uniformly, we're observing anti-conservative behavior. Our calculated family-wise error rate exceeds the desired multiple testing significance level. It is somewhat interesting here that the step sizes don't seem to impact this family-wise error rate, and I'll comment on this later. So any statistician would say, okay, this is not good. We don't want to have an anti-conservative hypothesis test. So we dove deeper into why we were making an anti-conservative scan and say, how bad is this actually? So I computed the minimum of the IVD rates in windows of size 3. And then for those windowed minimums, I saw if the maximum over all those windows exceeds our threshold. And what happened was that our bars go down, and in many cases, they're now below our desired significance level. What this tells us is that when we were making these type 1 errors exceeding our threshold, we were just barely exceeding it for a small 1 or 2 steps along the genome. The sort of recommendation to any users of our method then are that you should just be slightly skeptical of results that marginally pass our significance level. What is a little bit interesting is that we did appear to appropriately handle the step size delta. And this is practically important. So if you choose a smaller delta, you can get more precision in locating selected loci, putatively selected loci if they're out there. So in the literature, we have the Palomar 2018 paper that uses an ad hoc spacing. And their spacing is similar in size to our spacings. And then we have Chen et al. Who use an ad hoc spacing that's very large. And so in that case, they might not have a lot of precision to identify any associations they find in their study. We're adjusting naturally in our method for genome size, whereas this 5e to the -8 level was based on the human genome size. And a caution is that the choice of delta, while it has this trade-off with the precision and locating selection, this will impact the number of tests, thus what the threshold is, and the power to detect weaker forms of selection. We did power simulation. So we did some simulations with our algorithm and incorporated selective sweep selection coefficients. Here are the results. The different bar charts, the different bar plots give us different frequencies in the current day of the sweeping allele. And we're starting to really gain power around this 0.01, which some or many population geneticists might refer to that selection coefficient as a moderate to large selection coefficient. On the left, I was showing you the selection scan with 2 centimorgan threshold for the IBD detection. On the right, I'm showing you a selection scan for 3 centimorgans. And I'm not actually hiding data here. The plot is empty because we had no power. And our ultimate recommendation here is that you should use the 2 centimorgan scan. It was anti-conservative, but it has more power. The 3 centimorgan scan was less anti-conservative, and that was consistent with our central limit theorem, but it had no power. Now I have some real data analysis results. So this is our selection scan in 3 different ancestry groups: European, South Asian, and African. On this slide, we're returning to our initial analysis. So this will be our common figure here. We've got IBD rate on the vertical axis, the base pair along the genome on the horizontal axis. The analytical method in the legend will be our multiple testing. The heuristic is the prior 4 standard deviations method. And in this scenario, we actually have this orange heuristic line and the green analytical line right on top of each other. So it's hard to distinguish the slight difference. One would say, okay, we actually got pretty lucky in our initial work. I'll say that this did replicate in white British self-report samples from a United Kingdom biobank, and it replicated in different sample subsets in both the British biobank and the United States biobank. Some of these loci, so we have LCT again, which is believed to be one of the largest signals in humans, giving dairy tolerance to digest that late in life. These signals on chromosome 6 and 12 are involved with immune response. And then the signal on chromosome 15 is involved with pigmentation. And these immune-related phenotypes and pigmentation-related phenotypes are often discussed in the European selection literature as one of the main drivers of possible sweeps. Now I have the results for our Indians who self-reported as Indian in the UK Biobank. Again, the thresholds are on top of each other. We do see the signal at LCT, which is a result that's been consistent with existing literature that says that there could be a haplotype that is identical by descent with the haplotypes in Europeans that have that selected allele. And I'll highlight a signal on chromosome 16. It contains a single gene which is involved in the early development of skeletal cells. Why I'm in particular highlighting this is in the following slide. So now these are the results for our selection scan of African ancestry samples in a United States biobank. One thing to note here is, finally, the analytical method is higher than the heuristic method. This is pointing out the importance of having a principled way to derive your family-wise significance level. In this case, our estimates of the African ancestry gave higher thetas, which was saying that the tests were less correlated. So a larger correction for that. And we see a very large signal at this skeletal loci on chromosome 16. We thought this is quite interesting at this point. There haven't been a lot of selection scans in African samples, and at least in one we're aware of, they didn't really find any convincing evidence. We also will note that this pattern of having, say, an excess IBD rate or excess haplotype structure across different populations has been indicative of something that might not be a sweep, but could be, say, balancing selection, or other evolutionary mechanisms. And we have replicated this result in self-report samples of Black British in the UK Biobank. Just a brief review of our work modeling selective sweeps using identity by descent segments. This was a preprint, and it should appear in the American Journal of Human Genetics very soon under this same name. So recall, we have our coalescent under selection. On the left, we'll have, say, a weaker selection coefficient, and holding the time of the selected variant constant. On the right, I've increased the selection coefficient. And the effect of this is that maybe in the current day, we have a higher probability of having more samples that have the selected allele, and then their recent common ancestors are more recent. And so we might expect to observe more long detectable IBD segments, and that's the signal that we've been studying. Some updates, and they've mainly been focused on the selection coefficient estimator. So we compared that quite favorably to some other methods in the literature. One tries to solve or optimize a likelihood for a learned coalescent tree. We think the issue here was trying to infer the tree, and issues perhaps that the tips of the tree could bias the downstream analysis. And then we analyzed a neural network method that simulated data under selection coefficients, and we didn't really find great results for that. We did some more empirical coverage simulations to say that overall our confidence intervals do get pretty close to the nominal or desired confidence interval coverage. And we did a simulation study that was indicating that our selection coefficient estimator is sufficient. That means that, albeit we dichotomized the IBD segment lengths, it wasn't actually useful to have the entire lengths. You just needed to know if they were detectable. And we have some comments in the thesis about robustness and interpretation under model misspecifications of time-varying selection. In conclusion, I, throughout this thesis, have developed a suite of methods and packaged them together as an entire workflow. And so the projects discussed here today are bullet point here 1, which is detecting selection based on an excess of identity by descent rate with a multiple testing correction that's aiming to control the family-wise error rate. In the general exam, we had some methods to estimate the frequency and location of an unknown and possibly ungenotyped selected allele. And conditional on those inferences, we estimated a selection coefficient. And we also provided confidence intervals for that estimator. And we've appealed in the thesis that the desirable coverage results we're seeing are following from our central limit theorem, and then a delta method, which you can see in the Casella and Berger statistics textbook, appealing that this might work quite well when we have forms of weak selection. And this is all put together in a Python package that I call iSweep. It has about 4 different bioinformatics pipelines in there, such that these days I run a single command on the terminal and it does my entire analysis. So that's exactly why we've been able to replicate so many results in these different human populations across different data sets. Thank you all for attending this talk. This is the end of content. I just have some brief acknowledgements. So first, to my committee chairperson, Sharon, who's been sort of my main advisor and has imparted much research and statistical advice to me over these past 5 years. Professor Elizabeth Thompson, who was a co-author with me on our first project about the central limit theorem. My committee members, Kelley Harris and Amy Willis. Another co-author, Ryan Waples, has helped me build a lot of intuition for PopGen in our discussions, as well as the lab members, Roy, Nobu, and Robert. Some other mentors in statistical genetics, Professors Elizabeth Blue, Ellen Wiseman, and Tim Thornton. And then other members in the statistics department, our office staff, our advisor Ellen, Daniela Witten, and Emma Perkovich. So yeah, thank you for making it through this Zoom talk.